Thank you again for joining in for the first MPG primer of the season. I'm Diane Genero, and together with Sarah Morton, we are co-organizing the primer series this year. Again, thank you for coming. This year we are kicking off the primer seminar series with a talk by Dr. Robbee Wedow, who's a postdoctoral fellow in the Robinson Lab at Broad. He's also an affiliate of the Harvard Department of Sociology, where he focuses on social science genetics with particular attention to the intersection of social factors, environment, and genome. Robbee has previously used the genomics of same-sex sexual behavior, studied the genomics of same-sex sexual behavior, self-perceived weight status, and tobacco and alcohol use. In addition to his research at Broad, Robbee teaches courses on research methods in social science genetics at Harvard. His research also focuses on ethically engaged scientific communication. And Robbee, we're very grateful to you for joining us for this talk today. I also wanted to mention that Robbee has kindly agreed to answer questions at the end of the talk. And so please be sure to post those. I know that Sarah is going to post some more information in the chat, so you'll have a sense of how to do that. And at the end of the talk, I will go through those questions and then mention the names of people who have posted specific questions in hopes that they will be able and interested to voice those questions. So thank you again, Robbee, and please get started when you wish.
Okay, hi everyone. It's great to virtually see you all today. As Diane mentioned, I have many affiliations here at the Broad Institute, but also in Boston, but my work primarily focuses on the intersection of complex trait genetics and sociology. So I'm really pleased to be presenting here today, although maybe perhaps not so pleased to be sharing on the topic of the dark history of eugenics, but we'll get started. Before I begin, I think it's really important to note a few pre-talk items when we're talking about a topic as complicated and sometimes negative as the history of eugenics. Let me start first by giving copious thanks to a few people that helped contribute slides to the slide deck I'll be sharing today. First to Ben Neal and Leah Davis. Together they organized a slide deck for a talk that was presented during the STEM, Strike for Social Justice, to the Analytic and Translational Genetics Unit about a month ago. Also thanks to slides from one of my graduate school mentors, Greg Carey. And also finally, a few thank you notes to the newly formed Analytic and Translational Genetics Unit Justice Initiative for some of those members' comments on this talk as I created it. So I think it's fair to mention the very sensitive nature of a topic like the history of eugenics. It's really critical, I think, to be talking about topics like these in our community, especially now, given the increasing civil unrest and xenophobia that's going on in the United States, unfortunately. So together during this talk, we'll be traversing a difficult and even often viscerally revolting history that may affect different individuals in different ways. And so I just ask for lots of respect when it comes to this topic in the Q&A and the discussion, and then also in the following larger connected MPG session that happens after this talk. As a community, we all make mistakes and learn and grow from one another, and that's one of the wonderful and beautiful parts of being in a scientific community like the Broad. I also want to acknowledge my positionality and privilege as a white male presenting on a topic that often affects minority populations. I, of course, have never been the direct victim of eugenic thinking or racial discrimination, so I think it's just important to acknowledge that up front. And finally, the goal of both this primer and the MPG session, the connected MPG session that follows, is to provide a background for future discussion and to take what I think are important first steps towards creating a more fair and anti-racist environment here at the Broad and in our own research groups, and also hopefully for larger science. So with that said, I sort of invoke the muses, if you will, to help me use the slides and my knowledge to contribute to this goal. And then another sort of pre-talk slide, why talk about the history of eugenics in the first part, or in the first place? I think it's really important that as geneticists, we acknowledge that much previous work in the genetic space is grounded on sort of a shaky pseudo-scientific belief that there in fact exists empirical biological evidence that can be used to justify some individuals or groups as inferior. And so of course this isn't correct, and we shouldn't forget the history, but we also need to be salient of what social scientists have been referring to as the backdoor to genetics. And so it can be slippery at times for some people, especially people out there outside of the scientific community, to slip back into this eugenic thinking. And that makes the history of eugenics, both in the historic timeline and the social conscious, perhaps not so much ancient history at all. It's very much alive. And again, in this historic time of civil unrest, I think it makes it all the more important that we keep our history as geneticists in mind as we do our work. Another important thing is that I think that rather than assuming that our data speaks for itself or can be, interpreted in an unbiased, fair fashion, we have the duty to consider the larger language, context, and framing of our work so that we can do our best to help ensure that our work helps others instead of harming them. Okay, so now I'll jump into the larger part of the talk on the history of eugenics. I'll begin, as most talks on eugenics do, with the notorious character Sir Francis Galton. So eugenics at its conception, and at the time, was thought of as a modern project for helping the human population or the human race through selective breeding. This way of thinking was developed by Galton, who was a statistician, a psychologist, and a biometrician. He began much of his thought in the 1860s, and he was inspired by his half-cousin, Charles Darwin. So he was inspired by Darwinism and its theory of natural selection, although Darwin himself didn't necessarily agree with the conclusions that Galton drew. He read, of course, on the development of plant and animal species and desired to apply it to humans. And he became concerned about which heritable traits were spreading through the human population. And then in the year 1883, Galton named his research formally eugenics and started to posit a system of policies to, quote, rationally improve biological quality by controlling reproduction. You can see there under the picture of Galton, the definition of eugenics is the study of the agencies under social control that improve or impair the racial qualities of future generations, either physically or mentally. Okay, so just highlighting kind of what the scope of the history of eugenics looked like. When we think of eugenics, we think of the bulk of eugenics and eugenics thinking occurring especially in the United States between the years 1900 and the 1950s, although of course it went well beyond World War III in many places and persists in some part today. By the time we sort of had full-blown eugenics thinking going on in the United States, there were over 30 different countries that had all sort of developed their own versions of eugenics and also connected policies aimed at things like reproduction. At the time, it was considered politically progressive. So it was sort of afforded as the use of rational and science to improve the human race or the human population. And it involved organizations of social formers working together with scientists, geneticists, and doctors. And it was often called the science of human heredity at the time. There were two different sort of arms of eugenics: positive eugenics, in which individuals that were, quote, fitter were encouraged and educated to have more offspring, while with negative genetics, more unfit, quote, unfit individuals were persuaded, pressured, or compelled to have no offspring, not to pass on their, quote, defective genes. And unfortunately, this often involved the use of compulsory sterilization. So the unfit, complex, subjectively defined characteristics that were targeted for elimination were such loose things described, or such loose traits like criminality, epilepsy, bipolar disorder, alcoholism, Feeble-mindedness, a question you'll hear a lot, or a phrase you'll hear a lot in this talk, which was kind of a catch-all term used at the time to describe varying degrees of mental disability or learning disability, with the idea that moral defects led to crime and laziness, and individuals with these traits became a financial burden on the normal population that should be prevented from passing on their unfit genes and qualities. Environmental factors like poor housing, poor nutrition, inadequate education, discrimination, etc., that might have an important influence on these traits were often dismissed in eugenic thinking. So another notorious eugenics thinker was Charles Benedict Davenport, who was a Harvard zoologist, a taxonomist, and also someone who studied the heredity of albinism and neurofibromatosis. He was heavily influenced by Galton and also Galton's protege, Carl Pearson, who was a statistician and a mathematician. He co-edited an influential journal called Biometrica, and in 1911, he published an influential book called Hereditary in Relation to Eugenics. Over time, Davenport became a Mendelian, turning towards strong belief in eugenics and also genetic determinism. And maybe most importantly for the US context and the spread of eugenics in the US, he became the director of Cold Springs Harbor's labs in 1904, where he founded the Eugenics Records Office in 1910, and then moved much of the research that was going on in eugenics in the United States to Cold Spring Harbor's lab around that time. So here was Davenport's eugenics creed. Like when Ben presented this talk at the ATGU talk, we spent some time sort of reading it and leaving it on the screen. I'm actually going to read the creed because I think it really reflects some of the thinking that was going on for Davenport, but also some of the thinking that was going on and beginning to spread through society at the time. Okay, so the creed starts with, I believe in striving to raise the human race to the highest plane of social organization, of cooperative work, and of effective endeavor. I believe that I am the trustee of the germplasm that I carry, and that this has been passed on to me through thousands of generations before me, and that I betray the trust if that germplasm being good, I so act as to jeopardize it with its excellent possibilities, or from motives of personal convenience to unduly limit offspring. I believe that having made our choice of marriage carefully, we, the married pair, should seek to have 4 to 6 children in order that our carefully selected germplasm shall be reproduced in adequate degree, and that this preferred stock shall not be swamped by that less carefully selected. So beginning to hearken to some ideas of fit genes being passed on in fit marriages with fit individuals. I believe in such a selection of immigrants as shall not tend to adulterate our national germplasm with socially unfit traits. And finally, I believe in repressing my instincts when to follow them would endure the next generation. Okay, so another important figure in the history of US eugenics is Harry Laughlin. Harry Laughlin was a high school teacher and eventually a principal in the state of Iowa who had his PhD in cytology. He had some background in animal breeding and was interested, like Davenport, in eugenic applications to humans. And so through correspondence with Davenport, he was invited to become director of the Eugenics Records Office at Cold Spring Harbors in 1910. And at this time, of course, this is when the ERO became the epicenter of genetics research and policy development. And that office was operational at Cold Spring Harbors from 1910 to 1939. So what did sterilization, compulsory sterilization, look like in the United States? By the 1930s, 30 different US states indeed had forced sterilization laws on the books. So you can see on that chart there, or that map of the US, that the dashed lines or the dashed states and the black states are the states that eventually had, in the 1930s, compulsory sterilization laws. The white ones are the ones that didn't at the time. And this allowed the state to enforce sterilization of words you've already heard: feeble-minded, insane individuals, criminalistic, the deaf, deformed, and also dependent individuals that included orphans, ne'er-do-wells, tramps, the homeless, and paupers. Unfortunately, over 60,000 individuals were legally sterilized under these laws in the US through the 1970s, and sterilization continued in the US in some capacity all the way up into the early 1980s. Another character for you, another notorious character, was Henry Goddard. Henry Goddard was the director of research at the Vineland Training School for Feeble-Minded Girls and Boys from 1906 to 1918. And one of his biggest claim to fame in the eugenics movement was a translation of the Binet-Simon intelligence scale and a resulting classification for feeble-minded individuals. So he came up with a three category classification. The first one was the classification of moron with the relevant IQ ranking, as you can see there to the side. Imbecile, and then the third classification was idiot. You can see there's sort of a propaganda-like diagram drawn at the time. That might actually even be from an academic publication of one such interpretation of the scale that Goddard came up with. He believed that morons were unfit for society and should be institutionalized, sterilized, or both. Goddard also was the first one to use the word moron as a clinical term in the clinical setting. His work was notorious. He established many testing programs that took off around the US for classification of the feeble-minded, and he also famously started some testing programs on Ellis Island to identify feeble-minded individuals in the immigrant community as well. So beginning, in the 1910s and continuing up through the 1950s in the United States, eugenics thinking began to sweep through both American culture and society. You can see here an image of some men holding signs that say things like, "I must drink alcohol to sustain life. Shall I transfer the craving to others?" Or, "I cannot read this sign. By what right have I children?" And it sort of continued. There was propaganda as the movement continued in the United States. Famously, the American Red Cross began to play a role in what you may have heard of as fitter family contests. So as the eugenics movement swept through the United States, it became quite popular to put on demonstrations of eugenics and fit families, especially at state and local fairs. So in these fitter family contests, they would essentially administer psychological and physical exams to family members and also collect pedigrees. And then family members would be given an overall letter grade of eugenic health, and the family with the highest grade would be awarded a trophy or a medal like you see there. These became signs of great pride for the individuals that were deemed to be sort of the most fit families, those most encouraged in the population to continue to fit on or to pass on their fit genes to the next generation. Unfortunately, eugenics also had a deep impact on immigration and immigration policy. Some of the most famous cases came from testimony that was given by Laughlin. So there was a 1920s House Committee on Immigration and Naturalization where Laughlin provided expert testimony on excessive insanity among Southern and Eastern European immigrants. That was later reviewed. It was later reviewed and showed that he relied on alternative facts or badly misrepresented and manipulated data. There was also an Immigration Act in the United States called the Johnson-Reed Immigration Act of 1924, where he argued for extreme vetting of unfit immigrants. This law primarily aimed at further restricting immigration of Italians and Eastern European Jews and Africans, and outright banned the immigration of Arabs and Asians. If you look there, you can see an excerpt from some of his testimony where he's essentially saying that we should be giving examinations to immigrants in a similar way that we do for the viability of stock and plant, or for stock with plants and seeds. Sort of a very notorious act in the United States happened in the state of Virginia. So Virginia was a state in the eugenics movement that had particularly controlling eugenics policies and sterilization policies. So the Virginia Racial Integrity Act of 1924 used genetics as its basis. It made it illegal for whites in Virginia to marry outside their race, and it narrowly defined who could claim to be a member of the white race, stating that the term white person shall apply only to such a person that has no trace whatever of any blood other than Caucasian. There was also a Pocahontas clause where those with 1/16 or less of the blood of the American Indian were also counted as white. This was on the books for a while and was finally struck down in the Supreme Court via the Supreme Court ruling Loving versus Wade in 1967. Another historic Supreme Court case that you've probably heard of is the Buck v. Bell case. So there was a colony called the Lynchburg State Colony for Epileptics and the Feeble-Minded that housed many of Virginia's poorest residents, especially teens from broken homes and those whom state officials considered socially inadequate. Carrie Buck was one such resident of the colony and was sent to the colony because she was poor, pregnant, and also because her mother was institutionalized. So there you can see a picture of Carrie Buck and her mother. At some point, Carrie had an impending sterilization, and she was chosen to be used as a test in the Supreme Court of the constitutionality of forced sterilization in Virginia and in the United States. And so the testimony was quite harsh. In that testimony, individuals testified that her promiscuity was evidence of her feeble-mindedness, even though in truth she had been raped by the nephew of her foster family. One of the notorious Supreme Court justices who was part of this case was Justice Oliver Wendell Holmes. And when he wrote the decision, the decision on this case that in fact upheld the Sterilization Act of 1927, and by which Carrie Buck was eventually sterilized, he has a very sort of notorious quote, and that quote is, "It is better for all the world if instead of waiting to execute degenerate offspring for crime or to let them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their work. 3 generations of imbeciles are enough." Of course, eugenics didn't just take place in the United States, it took place in lots of countries. The most notorious of these places, of course, was surrounding eugenics and Nazism in Germany. There's lots to talk about, but just a few summary points on this slide. In 1920, Karl Binding, who was a jurist and a legal scholar, and Alfred Hoche, who was a German psychiatrist, used eugenic thinking in a book that became very influential called The Permission to Destroy Life Unworthy of Life. This phrase, life unworthy of life, became actually a social category that unfit individuals were sort of placed into that were then subjected to the atrocities of the Nazi regime. In 1924, Adolf Hitler published Mein Kampf, and in that book, he actually uses the US eugenics movement to uphold some of his ideas. So he writes, "There is today one state in which at least weak beginnings toward a better conception of immigration are noticeable. Of course, it is not our model German Republic, but the United States, sort of hearkening to some of those eugenic policies that he found favorable, especially towards immigration." And then in 1936, the University of Heidelberg gave an honorary doctorate to Harry Laughlin for his work in the science of racial cleansing. And unfortunately, at the end of Nazism in Germany, over 400,000 individuals that were considered hereditary defectives had undergone forced sterilization, with over 200,000 adults and children with disabilities being killed via euthanasia. And of course, the racial cleansing of the Jewish population in Germany and around Europe was perhaps one of the greatest atrocities of Nazism in Germany. So what happened after World War II? This was a time when we began to reflect on what had happened in World War II and also in part the eugenics movement. So this is the time when major academic centers began to shut down eugenics programs. The science was debunked, or the pseudoscience was debunked at this time. And this is also the time when protections for research subjects began to grow in part. So in 1949, there was the Nuremberg Code, which in Germany established some sort of informed consent, saying that research must be for the good of the public, must be justifiable based on previous work, must minimize risk, and must stop if the risk becomes too great. There was also the 1964 Declaration of Helsinki by the World Medical Association that established the right to self-determination and the right to make informed decisions, noting that it's the investigator's duty is solely to the subject and that the subject's welfare must always take precedence over the interests of science and society. There was also the 1978 Belmont Report, which was written by an organization formed by the National Research Act of 1974 that established respect for persons, so protecting the autonomy of all people and treating them with courtesy and respect and allowing for informed consent without deception. The principle of beneficence, or the philosophy of do no harm while maximizing benefits for the research project and minimizing risks to the research subject. And also justice, or ensuring reasonable, non-exploitative, and well-considered procedures are administered fairly and equally. And of course, many of these early sort of formulations of protections for research subjects went into the development of what we know today as the IRB, where experiments are justifiable and scientifically sound, where there's informed consent, and where there's review of things like conflicts of interest and assessment of risk and benefit. Forced sterilization programs did not end totally in the United States, as I mentioned, until around the year 1980. So that's very recent in the historic record. Many ended by the 1960s, but women of color were still targeted by state clinics up through the 1970s. And unfortunately, it's estimated that as many as 25 to 50% of Native American women were sterilized between 1970 and 1976. A few other mentions of major ethical breaches that are quite famous, but are worthy of mentioning here in the context of eugenics. The first was the Tuskegee syphilis study, which I'm sure most of you have heard of. Those trials went on from 1932 to 1972. They were conducted by the United States Public Health Service. The purpose was to observe the natural history of syphilis. But the end result here was that medication was withheld from the African American men in the study even after treatment became available. Obviously, a big ethical breach there. The Jewish Chronic Disease Hospital, in 1965, elderly hospitalized patients in Brooklyn were told that they were in a research study, but were not told they were being injected with live cancer cells to study the mechanism of immune reaction. And another famous incident, the Willowbrook Hospital study, 1972, mentally disabled children in an overcrowded state institution with really deplorable conditions on Staten Island were deliberately infected with hepatitis to help researchers seeking an effective preventative vaccine. There are many other examples. Those are just a few I highlighted here. Okay, so given the history, I'll spend a few concluding slides here talking about our responsibility. These thoughts come not only from me, but from some others as well. Unfortunately, the idea that some groups are biologically inferior to others tends to sometimes creep back into the public conscious. And again, in today's political climate and civil unrest, I think that that there's a slippery slope sometimes back into eugenics thinking. And so, just yesterday, we hear of allegations from a nurse, and I think as of this morning when I listened to NPR, up to 17 immigrant men or women about unwanted hysterectomies and gynecological operations that left them sterile in an ICE immigrant detainment facility in Georgia. And so I really think that it is of the utmost importance that in our work, we communicate culturally sensitive topics responsibly and widely, anticipating the potential implications of our research. For me and my work, when I'm working on sensitive topics that have the potential to have impact for communities, this for me calls for a new framework for communication and outreach. And for the careful eyes of diverse interdisciplinary teams of scholars working together with geneticists and biologists, but also with advocacy organizations and community members, especially when the work that we do has the tendency to affect different members of different communities. I think that good science is also well-communicated science. And so, I urge us to try to be as proactive as possible. I think that it's time where we move from the culture where scientists are just in the science lane and are not necessarily thinking about policy or downstream ramifications. So, I think sometimes this will call for scientists to have to go out of their comfort zone to ask about why we might want to do research in a particular area, within limits, of course, because research isn't designated from the start, but to also ask questions about what is the end game and what are the potential impacts for communities that I might be studying. It's also, of course, important to remind ourselves as often as possible that studies should yield data that is sound. We should be engaging in studies that are well-powered, that are replicable. We should be doing our best to engage in data sharing policies, open science as much as possible. This isn't just because it produces the best empirical results in our science, but also if you're doing well-powered studies that are replicable, I think that has a certain level of protection against the sometimes pseudoscientific thinking that, for instance, crept in the eugenics era. And then, when it comes to my experiences doing outreach and communication, don't hype the results as much as possible. Be careful with press releases and media coverage. If your work does get picked up in the media, as much as possible, try to seek out responsible scientific journalists and follow up with media error corrections. These are all sometimes very difficult things to do, but there are steps that we can take as a scientific community to try to be proactive in the communication of our science. This is one short example from some work that I did with many others when we looked at the paper on the genetics of same-sex sexual behavior. You can see here that we tried our best as a scientific team to engage limitations and cautions as often as possible, and to create things like websites with FAQs and shareable material to make sure that we were clearly and effectively communicating, what the study meant, but also, and maybe even more importantly, what the study didn't mean, especially for lay audiences. Okay, so this history of eugenics that I've shared, or the history that, any negative history of science, shouldn't let the past stop our current scientific efforts. Most scientists, especially at the Broad Institute, most geneticists are out there to try to do good, to help others, not to cause harm. But I do think that we should remember that the genetic science that we engage in can be easily misinterpreted, or sometimes even unfortunately co-opted to forward messages of biological determination or inferiority. I'm thinking here about things that are easily misinterpreted by the public, like gene editing technologies, what's actually going on when we say we're editing the human genome, polygenic scores, especially when it comes to terms like prediction, or when polygenic scores are applied to behavioral outcomes, or even something that seems as obvious to a geneticist, that genotypic data cluster by ancestral group. How do lay audiences or minority groups interpret what it might mean that genetic data clusters by ancestral group? And just the final slide here, I think that it's important that we revisit the past of eugenics as a reminder of both the importance of conversation around really sensitive topics around this, but also as a reminder of the importance of what it means to do good, sound science that will help instead of harming, and that we also can use this past as a reminder of the impact that our science can have in order to think of, or as a reminder of the impact that our science can have, and also as a way to think about the language, historical context, and framing when we're out there in the world speaking about our science, both within our groups and to others. Okay, and that's it from me. Thank you so much, everyone.
Thank you very much, Robbie. We really appreciate your talk. Just by way of reminder, I know that Sarah has posted about asking people to raise questions. There are 2 ways to do that, and I see that 3 people so far have posted questions in the Q&A, which I will voice in just a second. You're also welcome to use the raise hand function if you would like to raise a question yourself, and I will do my best to call on people in order there as well. Robbie, the first question came up quite early in the talk, and I will just voice it now. And the question was, wasn't LGBTQ included among the unfit in some of these areas that you described?
Yes, so definitely LGBTQ individuals would have been included in the unfit. Sort of, the definition and change of sexual identity over time may have identified different groups differently in those categories, but yes, LGBTQIA+ individuals were in some countries and some policies included in the unfit. Yeah.
Thank you. The next question was, do you think that the post-war rejection of eugenics would have happened were it not for the Nazis' embrace of eugenics?
So, that's an interesting question. I think that it was the most salient and most notorious example of the use of eugenics, and probably the thing that triggered us to begin to really look back on those policies. I would like to think that at some point it would have been debunked and that we would have begun to do better science. But I also think that it's likely that the eugenics movement would have continued its slower climb through the world had it not been for this particularly atrocious example of eugenics. So I think eventually it would have gone away, but I think it could have been a slower timeline potentially. Not saying that the embrace of eugenics in Nazi Germany was good for that reason, just saying that I think it accelerated or catalyzed the decline of eugenics.
Thank you. I just want to interject a question. Also, you described toward the end of your talk some important responsibilities that scientists have for sort of keeping careful attention to how their stories get told in the popular press. And to me, it seems like one of the many challenges in that regard is that a story can be picked up and very carefully related by a scientist in its initial rendering in the press, and then kind of get changed as it gets told repeatedly. And I wonder what sorts of paths or preventative measures, or I'm not even sure quite the right word here, but you might advise for scientists to try to keep as best they can sort of a role in making sure that story gets told in all of its complexity and with the care that's necessary.
Sure. Yeah, so I do have some experience working with different teams of scientists, especially regarding the work that I've done around the genetics of educational attainment and the genetics of same-sex sexual behavior. So, first, that note that I gave about being proactive, thinking about what you might want to go into a press release if you think that the paper or that the work is going to be picked up, potentially identifying sources that would be good to share early drafts or versions of the work with who you might not or might know might be more responsible. And then, depending on your level of engagement, I think it's really critical to try to make sure that media errors are corrected. So you can ask for corrections. You can respond on blog posts if you'd like to. And then if the science that you use ends up getting misused in some degree, you can try to work together with your research team to draft a response or to put something out there to sort of push back against it. So it's sometimes quite difficult to do this, but I found that in my own work, when you sort of have a unified strategy in place and you sort of go after errors in the media or something like that, you can make at least some headway toward correcting or putting the more factual interpretation out there.
Thank you very much. There's a set of questions that are arriving just now. One of them that came up a while ago is about the possibility of a deeper connection between eugenics and Nazism and the treatment of Eastern European populations such as Slavic nations, and whether there is perhaps also a connection with the church. I know you talked about elements of that history.
Yeah, so I'm unfortunately not aware of this particular example or case, so I don't think I have the insight to comment on this question, unfortunately, although I'm happy to follow up with a researched response or something like that.
Thank you. The next question kind of comes back to this point about scientists communicating about their work. I will read it now. How do you suggest communicating effectively and properly with regard to, on the one hand, we are often reminded that race is not biologically defined, but on the other hand, there are race-correlated differences in ancestry that do influence risk of disease, with sickle cell, of course, as an example.
Sure. Yeah. So this is obviously a tricky question. First, or a hard to answer question. But first of all, I think starting with the explanation that biological differences in ancestral or racial groups, especially when it comes to human variation, are actually quite small in general. Although there are some differences that exist, I think pointing individuals towards describing variation, human variation, as largely similar across individuals, that old saying that we hear often, is a really good way to start with that. But also reminding individuals that some differences, in fact, do exist, some small differences, and that our approach as scientists is that when these differences exist that show higher correlation in one racial or ancestral group, when they pop up, our goal as scientists is to use that information to better the lives of all humans and humans within those ancestral groups. So I think it's a tricky question that takes a lot of careful explanation when you're talking about race and biological race and ethnic differences, but one that just should be done with a lot of care and a lot of precision when you're communicating.
Thank you. And this question kind of follows up on exactly that point. At what point, and you've addressed this partially already, but at what point should this engagement start? So should it start at study inception? And what is your favored model of engagement for this?
Yeah, that's a great question. So yes, I think it should start at study inception. In my opinion, I think scientists should be thinking about the potential consequences of their research or ways that it could be misinterpreted. And so I think you want to start as early as possible, even if it just means sort of beginning to think about the ethics from the get-go, outlining different strategies or different or key important insights that you might take away from the study, and then drafting sort of responses with your team about how you might conquer some of those difficulties that may arise in the communication process. So my very favorite model is to start thinking about it as we did in the same-sex sexual behavior project, as the study was beginning and at the study inception. And then as you sort of continue through that process, you continue to adjust and adapt as other needs arise, and as you realize that you might, for instance, have to talk to more groups from the community or something like that. In many cases, especially when we're talking about topics that can be quite sensitive, this really does involve an enormous effort. And so it's good to be thinking about it from the beginning in a way that the team can readily adapt as you sort of go through the research process.
Thank you. Again, on this broad theme of engagement and representation, on the responsible conduct of science, what are your thoughts on how to increase the presence of BIPOC and other communities essentialized or underserved by genetics research? And this goes to the question of what gets studied by whom and how and for whom.
Sure. So, I think the way to do this is to begin in collaboration with as many diverse groups of individuals as possible. And so whether this means reaching out to diverse communities, one way that I think that this can be quite effective is to make sure that if you're studying research that, for instance, might speak to marginalized communities, that you're doing your best to ensure that you're reaching out to these communities as the research process unfolds, even trying to get them involved in the research process as much as possible. Also, we just need to be doing a better job as scientists at thinking strategically about how to get as diverse a group of scientists as possible involved in research at all levels. It has always been my experience that diverse teams of scientists make science better. And so, this is a big mission for all scientists and all research groups. One that I think is really beginning to take hold here at the Broad Institute, but we just have to do a lot better job making sure that individuals and communities that might be essentialized are involved not only in the research process, but a priori involved in how the research will be communicated, especially if it affects different communities.
Thank you. I also have a question about sort of public education, basically. It feels like much of the sort of misrepresentation of science and findings has to do in part with the public not having the depth of experience with thinking about the complexity of genetic questions as would be ideal, of course. And that's, of course, a really complicated set of issues because teaching is difficult and it's done in many different environments. I wonder if you have sort of a sense of how scientists who are working as scientists perhaps can collaborate with people who are working in more, explicitly teaching environments to try to deepen public understanding of genetics and the scientific process in a way that will help, people from a very young age to grow up with a deeper understanding of the process and perhaps a better ability to see the sort of the inappropriate melding of worrisome agendas and what is nominally, but maybe not actually scientific data.
Sure, yeah, so I've actually been thinking a lot about this lately, especially in collaboration with teams of science educators and educational scholars. I really do, from my personal perspective, I think that the way that genetics is taught in elementary education leaves much to be desired, often not even focusing on much beyond Mendel's pea plants. And so in terms of what we teach about genetics, I think a shift towards science education that tackles the complexity of human diversity rather than focusing on, for instance, deterministic sort of ways of thinking about genetics like Mendelian genetics and the pea plants would be very good. So I've been working on drafting ways to change the genetics curriculum in those cases with some of these individuals that I've been collaborating with. Also, in terms of the public, educating the public, this is very difficult and it's very hard to change ways of thinking that are salient in the population. But I think that one way to think about how we're going to do that is to think of those sort of tropes out there that should be taken down and to, whenever we can as geneticists, write clearly and signal sort of clear, easy to digest and understand ways to think about human variation and genetics instead of, for instance, describing something in a more complicated way. I've found that in my work, really clear, short, concise signals are the best way to go when you're thinking about sort of educating the public.
Thank you, Robbee. And I think that takes us to both the end of the list of posted questions and the end of the time for the seminar. We really appreciate your providing this introduction to this complicated and, as you said, in many ways troubling history. And we invite everyone to return in about 10 minutes for the MPG seminar, which will follow up on some relevant questions. So thank you very much, Robbee, and thank you again, everyone, for joining us for this talk.
Bye, everyone.
Bye. Thank you.